So now that we've gone over some of the history leading up to evo-devo, and we'll do some more on some of the early work of this with the Speeman-Mangold experiments in Unit 1.3. But I want to give you a big picture overview of some of the important modern techniques that we use as tools. Evo-devo is still a relatively young discipline, and not only that, but it really is multidisciplinary. We use a lot of classic molecular biology techniques. We also use a lot of new things that are just becoming available, and I wanted to review some. This is not an exhaustive list, but is a pretty good list of the techniques that are really important and central to the study of evo-devo. So we'll talk about each, give you a very brief overview, and then give you a quick idea of how they are used in the science of evo-devo. So the first thing that we obviously need to do when we are wanting to study where complexity comes from, how we move from a molecular level to the complex morphology, is we need to get into the genes, into the genetic material itself. And to do that, we need to be able to chemically isolate nucleic acids. So this is called DNA and RNA extraction. Both are somewhat similar. There are different methods, different columns, different chemistries that can be used to do this. One of the key ideas when you're doing a difference between DNA and RNA is the pH, but there are many different methods to do this, and we won't get into the details of all of them. I will give you an overview of kind of a classic phenol extraction of RNA. That's really where we want to start. We want to start at the RNA. So it's critical that you understand the difference between DNA and RNA. DNA, of course, is the entire genetic instruction manual, the entire genome of an organism, whereas RNA are only those portions of the genome that are being utilized, that are being expressed. So if I was to extract the DNA from your brain cells, hopefully you'd be dead or maybe a small sample, and extract the DNA from, say, a liver or a bone cell or any other cell with a nucleus, I would get basically the exact same outcome, right? So your DNA is the same no matter which cells I choose. Now, there are maybe some small mutations, particularly if you have cancers or viruses that have inserted themselves, and that's not a big deal usually. Sometimes they become a problem and spread, and those can be issues. But barring mutations, your genome is the same no matter what cell I choose. Whereas RNA is very, very different. So the RNA, the genes that are being used in your brain cell, is very different than the RNA in, say, a liver cell, right? So you might have a certain category, a subsection of those genes that are the same. Those are the genes we call the housekeeping genes. But then you're going to have a wide array of tissue-specific genes that are very, very different. Not only that, but RNA from a brain cell of a sleeping individual might look very, very different than the RNA from that same brain cell when that individual is awake. So RNA is the flexible, it allows us to react to our environment. It allows tissues and cells to change what they are doing from minute to minute. So RNA is really critical. And if we're going to now bring this into the evo-devo realm, think about this. What would the RNA look like in a developing leg cell versus a developing wing cell? If we're looking at, say, an insect, and we're looking at developing wings versus developing legs. And we might have some elements that are similar. They're both appendages. There might be some basic genes to grow an appendage that are going to be the same, but then we're going to get different genes, or at least genes working in different ways to make a wing versus a leg. So if we can understand the differences at the RNA level in different cells and different tissues, we really are beginning to understand why those tissues are different at the molecular level. A key step after we extract DNA and RNA is a polymerase chain reaction, commonly called PCR. Right? So PCR, I need you to understand this. It is fundamental to all molecular biology. This is maybe the most important reaction in all of modern molecular biology. So there are two areas that I want you to understand, and you might already have a good understanding of this. It is so common, you may have done it before, okay? But the first thing I want you to understand are the ingredients. And we're simplifying a little bit here, but we're getting all of the major important things. To do a PCR reaction, you need a sample of DNA that you're going to copy. And then you need primers. Primers are short, single-stranded pieces of DNA. that match a sequence on either side of your targeted DNA. And usually we're looking at fairly short pieces, 1,000 base pairs or less. But we want to target and we want to amplify and make many, many copies of that. So the primers serve as our beginning for our template copying. The next thing we need is a polymerase, and we use Taq polymerase. Taq is a shorthand for Thermophilus aquaticus, which is a bacteria that lives in hot springs. And the reason we use that polymerase is not that it is different in overall outcome. It still just copies DNA, but it is adapted to do it at very, very high temperatures. And so this allows us to manipulate the sequence of copying the DNA, which we'll look at in these next steps. So we use a polymerase from a hot springs bacterium. And then finally, we need nucleotides. Nucleotides are the basic building blocks for DNA, the adenines, thymines, guanines, and cytosines. And if we have all of those, and then the correct conditions, we add other ingredients just to make sure that everything is as close to physiological conditions as we can get it, then we are ready to begin copying our DNA. So we mix all of those in the proper proportions with our DNA that we want to be copying, and then we go through this reaction cycle. And it's a cycle because we do it over and over again. The very first thing we do is we heat to 94 or sometimes 95 degrees Celsius, close to the boiling point. Now, this would denature most proteins, but the Taq polymerase is adapted to these high temperatures, so it stays in its functional shape and works really well even at high temperatures. But the reason we heat it up is not to denature the protein. That would happen to if we had put human polymerase, for instance, and then the polymerase would no longer be functional. But we do it to denature the DNA. At these high temperatures, the double-stranded DNA unwraps and becomes single-stranded DNA. So that's step one. Step two we call the annealing phase. We drop the temperature down a little bit, but still rather hot. And this allows short sequences of DNA to anneal or stick to their matches. And so our primers find their most, and the reason this happens is it's just where they're most stable at that temperature, but they find their match and they bond. So we have short sections of double-stranded DNA on either end of our target DNA, but it's not cold, it's not cool enough to allow the entire long strands of DNA to completely re-anneal. So primers anneal at this temperature. We then heat it up back to a little bit to 72 degrees Celsius. This is the optimal temperature for our polymerase to work. And so at this temperature, the polymerase begins to make copies. And we make a copy of that strand. And we do this over and over and over again. And if we have 100% efficiency with each step of this cycle, sometimes we do less than 30, but typically 30 is about the target. But at each step, we double our amount of DNA that we make. So if we have one copy, after 2 cycles, we have 2. After 3 cycles, we have 4, and then 8, and then 16, 32, and so on. And so this is the power of geometric growth, where eventually after 20, 30 cycles, we end up with millions and millions of copies of our target gene. So that is PCR in a nutshell. You need to understand that again, both on the ingredients level and on the cycling level. So review that, make sure you know it. Gel electrophoresis is a very common and important technique also. Gel electrophoresis allows us to separate out DNA of different sizes, and there are other techniques that can do RNA or protein, but the most common electrophoresis is probably DNA. And so notice we have 3 different sizes of DNA in this column. We have a long one, that's probably the template DNA that went in, and then we have 2 pieces that have been digested or broken up. And so we can see different digestions lead to different sizes of DNA, and we can see that on the gel. This is accomplished most commonly by applying a simple current. DNA all has a negative charge, so if we apply a current, it's going to migrate toward the positive pole, and the rate of migration is dependent on the size of the DNA molecule. So small molecules migrate very quickly through the gel because they're little and can kind of worm their way through the matrix of the gel much more efficiently. And longer pieces of DNA take longer. So by applying an electrical current to a sample loaded onto a gel, we can separate it based on size. Okay, that is gel electrophoresis. The next technique we're going to talk about, and we're actually going to do this in 2 phases, is DNA sequencing. Now, at its heart, all DNA sequencing relies well, almost all of the modern techniques that we really use for large amounts of sequencing, they rely on PCR, that polymerase chain reaction at some point for some part of it. So the first one that we're going to learn about, by which most of the first decade of DNA sequencing, all of the data that was generated for the Human Genome Project, for instance, for sequencing bacteria genomes, all of it relied on Sanger sequencing. So this is how Sanger sequencing works. First off, we set up a PCR reaction, but there are two main differences to this PCR reaction. The first difference is that we only put in one primer. So rather than trying to sequence both of them, both strands, we are only going to be sequencing one strand. The downside of this is we don't get that geometric growth. We don't get 2, 4, 8, 16. Instead, we get 2, 4, 8, 10, 12. So we don't get as much output of it, but we have a single product that's being made rather than both strands. And that's critical, so we only are getting that signal from that one single strand that we are copying. The second major difference between DNA sequencing and our PCR is the fact that we add a small percentage of nucleotides that are defective, meaning that once they are incorporated into the growing chain, we cannot continue that chain also. So sometimes the Sanger method is also known as the chain termination method. But not only are they deficient in that they cannot be added onto and we can't complete the synthesis of that chain of DNA, but in addition to that, they also are fluorescently labeled. And so what ends up happening is, as we go through multiple cycles of this reaction, we end up terminating the reaction at different random sites. And if we do enough of them, we get every single site. And those terminated chains are labeled with a specific fluorescent marker. Now, originally, we had to set up 4 different reactions with 4 different markers because the markers were indistinguishable, so we needed them in different reactions so we could determine. They would then be run out on a gel, and you could read the sequence by looking at the order in which these different chains were present on the 4-column gel. Now, shortly after the development of this process, they developed a process where you could actually add all 4 reactions, all 4 chain terminations into a single reaction. So we could put them all in one reaction, run them out on a gel, and then differentiate based on which fluorescent marker was labeling the chain termination. So we could run them on one gel. And the outcome of this, I don't have a picture. I'm going to actually bring it in here. Let's bring one in. The outcome of this was what's called a chromatogram. I'll show you an image here, and I'll put this into the PowerPoint. Okay, so there are all sorts of different chromatograms. Here is one from a DNA sequencing reaction. And the different colors are representative of those different fluorescent molecules. So we could say, oh, look, we have 3 peaks here that correspond to the fluorescent wavelength for cytosine, what we've labeled cytosine. Here are 3 for thymine, then there's 1 for adenine, and so on. And so by reading these peaks, we can read the sequence of DNA. Now, each of these peaks just simply represents a absorption spectrum as that first short molecule comes out of the column. So there's our first absorption. It corresponds to C. Our second one corresponds to C. Our third one, and then so on. So we can recognize that DNA sequence. Okay, so we'll copy this and put it into the PowerPoint here. So our modern, kind of more advanced Sanger method would rely on these chromatograms. Now, this was used to sequence the human genome. We had to do millions and millions of different reactions and then line them all up and put them together. And it worked much better than the original 4-column, was more efficient, but was still very, very inefficient and took tons and tons of effort. So the first human genome was sequenced in about the year 2000. And it took hundreds of millions of dollars and thousands upon thousands of man hours and computing hours to do it all. Today, we have much more efficient methods. So I am also going to look at briefly the DNA sequencing method, which is often called next generation or modern DNA sequencing. And the most common, there are a couple of different variants that are competing for common usage, and some applications are slightly different, but by far the most widely used one still today is one that was developed by a company called Illumina. So this is the one that we are going to look at. And this allows us to do massive sequencing, just billions and billions of base pairs in one single reaction. And it's termed sequencing by synthesis. That's their term for it. So I've got a short video here I'm going to play. It's only about 5 minutes long. And I want you to understand the key components. I'll review them very briefly after we watch the video.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
Okay, so that gives you an overview. Let's shut this down. That gives you an overview of the process. Now, there's a lot of detail in there, and I don't expect you to know all of the details, so let's just kind of summarize it quickly. We take a sample of DNA and we cut it into small pieces. Then, after we get those small pieces, we put it on this chip, and they basically get stuck to the chip. They get amplified, so each little piece is now a cluster of DNA. And then we can actually watch, because of that, the fluorescent incorporation, the little burst of light that occurs each time a different base pair is incorporated, we can actually watch it occurring. And then this occurs on millions and millions of different clusters. And then we get all of this data back, and we have to assemble it together. Now, this technology is much, much cheaper per sequence that's being generated than the Sanger method. The Sanger method is still relied on for some small focused sequencing reactions, but this can give us an entire genome, whereas before it would take hundreds of thousands of man-hours and millions of dollars. Today, you can sequence an entire human genome and get back the raw sequence, and you can do it in just a few days for a couple $1,000s. And it's even getting cheaper and cheaper and more efficient all the time. So we are really entering a phase where we can generate massive amounts of data. Now, for this class, we are going to do this for transcriptomes. And in the first lab, we'll probably do some background on the organisms and what we're going to be doing. And then we'll do a little bit on an overview of the methodology. Okay, but just be aware of Sanger sequencing and those two key differences between Sanger sequencing and PCR, and then the Illumina sequencing. And Illumina sequencing still uses PCR and the initial phases to generate those random sequences of DNA at sufficient numbers to then be able to go on to the next step. So we will do a very quick overview of transcriptomes here. So cDNA refers to complementary DNA. And basically, there is a process using a reverse transcriptase that we can take RNAs in a cell and turn them into DNA. And this is important because all of the sequencing technologies are based on DNA. So if we want to sequence a transcriptome, we first need to take that RNA and turn it back into DNA. One of the methods of doing this relies on the fact that in eukaryotes, RNA has a poly-A tail. So we use a poly-T primer to copy all of the mRNAs, and then that mRNA becomes DNA. That DNA can then be put into the workflow. So here's our double-stranded cDNA, which is a DNA copy of the RNA transcript. This DNA can then be put into the same workflow for sequencing a genome. And so what we end up with if we do this is we get a transcriptome. And we're going to be hitting this a couple times. It's a really key idea, the difference between genomes and transcriptomes. A genome, we just start with the DNA, all of the DNA in a cell. And if we sequence it all and assemble it, we have the entire genome. But if all we want are the genes, or maybe even more critically, the genes that are being used in a particular sample, then we can use a transcriptome. We extract the mRNAs, we amplify and sequence only those mRNAs, and now we have a snapshot of the portion of the genome that is being used in that particular tissue type. And so that is a transcriptome. Okay, so make sure you're clear about the difference between a genome and a transcriptome. They're both important ideas and concepts, and make sure that you know what cDNA is and what makes it different from genomic DNA, right? So the cDNA is the expressed RNAs, mRNAs, that are being used in a particular tissue type. And it's different from the genomic DNA because it's only a small representative sample of the entire genome. It's only the genes that are being actively transcribed. Now, there are a couple of other techniques that allow us to visualize expression in cells. And there are two main ones that we're going to be looking at. The first is antibody staining. An antibody is a protein that is used in mammalian immune systems, and we can generate them to target a specific protein product. And in theory, the array of targets that antibody can respond to is incredibly wide. So for instance, if I take a fruit fly protein that I'm interested in studying and I purify it and inject it into a mouse, that mouse will have an immune response. It's not dangerous to the mouse, but even non-dangerous foreign things are, as mammals, our immune system responds to them and makes antibodies against them and gets rid of them, even if they're not dangerous. It's kind of a preemptive strike against any foreign entity in a mammalian body. So if we inject that fruit fly protein into a mouse, and then we wait for that mouse's immune system to generate a response, we can isolate antibodies that are specific for our target protein. We'll look at this in more detail in the lab, but usually there is a primary protein that targets the antibody. A primary antibody that targets the protein that we are interested in. And then we have a secondary antibody that binds to the primary and is fluorescently labeled. And the end result is a methodology that allows us to visualize where specific genes are being expressed. So in this diagram, red represents a specific gene that is being expressed in the trunk of this developing organism, but not in the thorax or in the head. This is a millipede, I believe. So in the mouth parts and in the head, we get very low or zero levels of expression of this target protein, and then we see it throughout the rest of the body. So that gives us clues, a beginning clue about what this gene might be doing. The second technique that we can use is an in situ hybridization. This also allows us to fluorescently label a very specific product. However, rather than labeling the protein product, an in situ hybridization labels the mRNA. The end result for development is basically the same. It's equivalent to an antibody stain. However, this can be much more specific to a specific individual organism and the gene that it's looking. Antibodies often are cross-reactive. So if you make an antibody for a specific gene in a fruit fly, it might also very well label that same gene in a spider or a cricket. Some of them are even broadly specific and will do many different phyla of animals. But an in situ is specific for just that one gene in that one organism, and it labels the mRNA. But the end result is still the same. We can see expression of a target gene with very different fluorescent labels. And in this one, we have 4 different fluorescent labels. Actually, just 3. Red, blue, and green. And they're all labeled differentially. Okay, so the process for in situ fluorescent hybridization, fluorescent in situ hybridization, sometimes called FISH as an acronym, is fairly complex. We'll do it in a little bit more detail in one of our labs near the end of the semester. So don't worry about it for now, all of the details of how it works. Just know that number 1, we're labeling the mRNA rather than the protein product, and it's more specific than an antibody stain. It's only going to do that one gene in that one organism. And then we need modern microscopy techniques to be able to visualize these things. So here is a diagram that incorporates both fluorescent, which is just using different wavelengths with different molecules to label things differentially. So I've got the nucleus labeled in blue, the cytoskeleton of this cell labeled in green, and then the mitochondria labeled in red. And these are a little bit more general markers, but we could also add on more specific markers that we're targeting one single gene. Okay? Confocal microscopy is a technique that allows us to filter out different wavelengths of light that are not in focus. So it gives us a clearer, crisper image by basically taking digital slices of the structure that we're looking at so we can see it in 3 dimensions. And we will do that in more detail in a lab for now, but just know that confocal is a way to get a crisper image without the out-of-focus components of a 3-dimensional subject. I'm actually going to skip microarrays. They're still used a little bit, but they're becoming less and less important. For now, all you need to know is they have been largely replaced by transcriptomes. By sequencing an entire transcriptome, we can accomplish much of what we can do with a microarray, and now that it's easier and cheaper to do it, many transcriptomic methods have replaced microarrays. I do want to spend just a little bit of our time, this is our last slide here, looking at methods by which we can alter gene expression. So far we've looked at how we can visualize it, see what's going on with genes being expressed, but we haven't changed anything. There are two main methods that are still used, and CRISPR is becoming very, very widely used and to some extent has replaced this first one, but this first one is also used. I won't worry about for now all of the details of how they use. I just want to talk about generally what they do and what they're used for in evo-devo. RNAi is short for RNA interference. And through a series of methods that have been, proteins that have been isolated from a various array of organisms, we can essentially turn off gene expression. Now, to be very technical, we're actually not turning off the expression. The gene is still being expressed, but as soon as it is being made, it becomes degraded. So we can basically, as soon as an mRNA is made, rather than going through and letting it, have a protein be made from that RNA, we can digest, break down that RNA so that even though it's being made, it's not being used. And so this allows us to essentially turn off genes and see what the downstream effect of that is. In a later class, we'll look at some of the steps along the way for doing that. But RNAi allows us to turn off or turn down gene expression. Now, CRISPR is a little bit more flexible. CRISPR works at the DNA level rather than the RNA level, and it allows us to, it's essentially a very specific and fairly efficient cut and paste mechanism, or copy and paste if you want to do it that way. Either one. Copy and paste mechanism for DNA. So we can go into an organism and into a cell, and we can change the DNA of that cell any way we want basically now. So it really is an incredibly powerful tool, and there's some ethical things that we'll investigate in a lab, in a later lab with CRISPR. We're not going to be doing any CRISPR in this lab, but we will talk about how it can be used for certain evo-devo techniques. So imagine you want to study a specific gene in a fruit fly, and you could go in and modify that gene, or maybe not the gene, but what if you modified the regulation of that gene so that that gene can no longer be turned on? And then you look to see what the impact of that is. So it's a really, really powerful tool that allows us to investigate the function of genes. All right, that's it for today. Some of the things that we talked about today, we just kind of laid a foundation. We'll talk about in the lab or in later lectures.